Hello everyone, this is Ketish here. In this video, I'm going to go through the AQA Biology Paper 2 Higher Tier from 2025. Question number 3 is on the topic of homeostasis and response. Tropisms help plants survive. What is geotrophism? Geotrophism is the plant's growth response. Is the plant's growth response due to gravity. Now, there are two types of tropisms, geotrophism and phototropism. Phototropism is the plant's response to light. Now, if you have seen the 2024 paper, you would have come across that they have actually asked a question on geotrophism. So here, they're actually asking a question on phototropism. So plan an investigation to find the effect of the direction of light on the growth of plant seedlings, which is called phototropism. You may use the equipment shown and any other laboratory equipment. So they have given you a lamp, a ruler, cardboard box, and cardboard box with slit in one side. Because they've said you can use any other equipment, you can assume that you've got more than one cardboard box. So how would you test or investigate the effect of the direction of light on the growth of plant seedlings? So for that, you would need plant seedlings, right? So let's say you have plant seedlings growing in petri dishes or in trays, right? And you want to grow the equal number of seedlings, right? And let's say we've got 4 of those dishes, and let's say I've labeled them as A, B, X, and Y. You'll see why I'm actually labeling it like this. So once you have the seedlings, you germinate them at the same time, so and expose them to the same conditions, so same amount of soil and all of that, so they're roughly about the same height at when you start. Now you would place the seedlings in the box, right, in each box, or you would actually take 2 boxes of this with the slit and 2 without the slit, and label one as A and then X, and label this as B, and actually hold on, it's the other way around. Let me just relabel that. So A and B for the ones without the slit, and X and Y with the slit. So ideally what you'd want to do is put these cardboard boxes, put A and X in a dark cupboard without any light, and you put B and Y where there is light, or you could actually place the lamp in front of the slit, and you'd make sure that you're placing the lamp at equal distance from the box, let's say 20 centimeters or so. So you place both boxes side by side, one without the slit and one with the slit, 20 centimeters away. You'd place the lamp so that both boxes are receiving enough light. Obviously the one which is covered is not going to receive any light. The idea is to find out if you place even with the slit or without the slit in total darkness, right? So you need one where you have total darkness, and you would place the box A and X there, and place 2 boxes with the light, and you put box B where it's fully covered, and Y as well, so that here in the total darkness, what you're trying to do is, even with the slit, if there is no light, what happens, and without the slit, what happens, and when there is light, what happens to the seedlings when it's fully covered, when it's not receiving any light, and when it's receiving light in just one direction. And you leave it for about a couple of days, and you periodically, every day, you could just remove it for a couple of seconds to spray water on the top of the seedling, so there's enough water, and then you place it back. You need to leave it for at least 3 to 4 days to make sure that the seedling is actually growing. At the end of, let's say, 4 days, right? Okay, before you do that, you would want to measure the length of the seedling. So at the start, length, and the end length, right? You'd measure the length, let's say it was 10 centimeters, and then after 4 days it's 20 or 20 centimeters. I'm just making up these numbers. And you'd also want to record the direction of growth. So is the seedling growing straight, or is it growing towards, or is it bent, and in which direction is it bent, towards the slit or away from the slit, and so on. And then you can actually compare. The reason you want to do this with 20 seedlings is so that you can actually measure the length of 20 seedlings and take an average reading. You don't have to do it in between the days. You just need to do it so that at the start and at the end, because the best way is actually to cut the seedlings after 4 days and then measure the length after you record the direction of growth. So that way you can calculate a mean value. Now there's a couple of things that I've already mentioned but worthwhile pointing. You have to make sure that you are controlling certain conditions. The conditions are actually controlled the exposure in terms of the boxes that the seedlings are placed. It's 20 seedlings in each box. Water them enough and allow them, all of them, to grow for 4 days, and make sure that the temperatures roughly are the same temperature as well, and so on. Here's a model answer. The method: place 20 seedlings in 2 boxes with slit and 20 in 2 boxes without slits, labeled XA, B, and XY. So I've actually used these labels to help me explain the next steps clearly. Place box A and X in darkness, and place B and Y in all round light or just one direction light. Leave the seedlings to grow for 3 to 4 days, only opening them to water them. Measure initial and final heights of shoots with a ruler. Straighten bent shoots before measuring. Record the direction of growth of seedlings, whether it's straight or towards the light. Calculate the mean height increase for each condition and compare the results. Now you don't have to discuss the results here. They're asking you to plan an investigation. So it's just the planning bit. The control variables are the same number of seedlings per pot or per condition, same temperature, same volume of water, same soil type or volume, same age of seedlings, same species or type of plant, and same time left for before and till the end. That should give you the 6 marks. For level 3, a method, you need to have a method comparing the growth of plants in light from one direction with plants in full light and with total darkness, along with control variables is required. Next question. Plant responses are controlled by hormones. Which hormones controls a plant's response to the direction of light? That would be auxins. Auxins. How does the hormone you named in question 3.3 control the plant's response to the direction of light? Now, let's say if this is a shoot. Right? And that's the soil. The auxins are actually produced at the tip of the shoot over here, and due to gravity it actually goes down. Now as it goes down, what auxins cause is cell elongation. So cell elongation. So it's not multiplying it, it's actually making the cells longer. So it actually, when the auxins reach the cells actually on both sides will actually grow longer, causing the entire thing to grow up. Now when light hits in one direction, what happens is the auxins actually move away from the light. When the auxins, especially to the shoot, when light hits on this direction, the auxins concentrate on the shaded side and actually will move to the shade side rather than the lighter side. It causes the uneven distribution of the hormone, which is the answer. So when there is uneven distribution of hormones, you're looking at the cells simply growing on this side longer, which causes the entire thing to actually just move in this direction towards the light. Which is the next question. Explain how a plant's response to light helps the plants survive. So they're not actually asking for how the auxins work. How does it actually help it to survive? So the leaves of the plant would receive more light, right? So there is more photosynthesis, which results in the production of more glucose. So more glucose is produced, right? Which is important for the plant to survive because it's the main molecule to provide energy for during chemical reactions. Now a quick note about auxins, right? Auxins have the opposite effect on the root cells, right? And this links to geotropism. So in a root, the shaded side contains more auxin and grows less, causing the root to bend away from the light. That brings us to the end of question number 3, and I will see you in question number 4.